Thank you very much, Emily, and thank you for the invitation and the opportunity to talk to you about our research. So my lab works on primarily cancer vaccines, as Emily alluded to. And as I'm sure most of you are aware, metastatic solid cancers, with a very few exceptions, are thought as incurable. However, the development of checkpoint inhibitor therapies, primarily the use of anti-PD-1 antibodies, have resulted in the induction of long-term remissions in some patients. And this is about 30% of the patients that are treated with anti-PD-1 go on to have long-term remissions. And the interest then has been in trying to understand why these 30% of the patients respond and why the other 70% fail. And what I'm showing you here is basically immunohistochemistry from a paper by Tony Rivas' group, where he was one of the first ones to do a correlate between immune infiltrates and clinical responses when you got anti-PD-1. And what was observed was that those patients that had responses tended to have what is called a hot tumor, and that is they were heavily infiltrated by CD8 T cells. The T cells and the tumor express both PD-1 and PD-L1. While the patients that went on to progress had what are called cold tumors, and that is there is very little T cell infiltration, and neither PD-1 nor its ligand PD-L1 are expressed. So one of the questions that then we have is, can we vaccinate patients such as these and initiate or reinvigorate their immune responses such that we convert cold tumors into hot tumors? So at the same time, the development of next-generation sequencing was allowing us to catalog the mutational burden of tumors. And this is from a paper by Alexander that was published in 2014. And what we see is that there is a spectrum of mutational burdens across cancers, with the pediatric cancers on the left side with very low mutations, less than 1 mutation per megabase of genome, and melanoma at the other extreme of this spectrum with a high mutational rate of about 10 mutations per megabase. Also, these mutations have the classic pattern of UV radiation, which is a C to T transition. So with this information in hand then, we started a project that we call the Melanoma Neoantigen project. And it was based actually in an all-observation 2 papers that were published in around 1990, mid-1990s, one from Terry Boone's lab and the other one from David Beach's lab, where they had actually reported that in patients that had cutaneous melanoma, you could see T cells that could respond to missense mutations. However, the process that it took them to identify these missense mutations was about a year because they had to use cDNA cloning. However, with the advent of next-generation sequencing, we could do this in a more time-compressed manner, and we could get to more mutations and a better and deeper analysis. So we came up with the hypothesis then that missense mutations arising from single nucleotide variants, somatic single nucleotide variants in tumors, could generate patient-specific tumor neoantigens, and that if we vaccinated, we could elicit T-cell responses to these variants. So we use next-generation sequencing to identify the mutation, and we develop a bioinformatic pipeline to make synthetic peptides. And then we use dendritic cells because our lab had already used for over 10 years autologous dendritic cells. In this case, dendritic cells that were mature with 4 stimuli and that were IL-12 producers. So I just said this. So this is the way our vaccine scheme for neoantigens it goes. So we get a tumor, and we also get normal. We make DNA from both normal and tumor, and we make RNA from the tumor. We use the normal DNA and tumor DNA to do exome sequencing. Then these are aligned using pipeline algorithms, and we identify those somatic mutations that are unique to the tumor. Then those are run through an epitope prediction line that is based on the patient HLA haplotype. We pick those that we think can be presented by the HLAs of the patients, and then we take into consideration genomic parameters and immunological information to pick somewhere between 7 to 12 somatic mutations that could encode for peptides that might be recognized by the immune system. We verify that those peptides can bind in vitro to the HLA of the patients, that they can refold HLA, and then those we consider validated candidates that then go into a vaccine. So this is just the name of the bioinformatic pipeline. And they go into an autologous dendritic cell line that consists of 3 doses that are 6 weeks apart over a period of 18 weeks. And what we also do is we immune monitor the patients for the generation of the immune response by weekly draws of blood that then are put in vitro and then are revealed by using multimers. So this is a representative analysis for one of the patients. In this case, this patient was HLA-0201 and HLA-B801, and we had 2 tumors from these patients. So both tumors were exome sequenced, and that allowed us to identify the number of exomic and missense mutations. So in this case, it was about 3,000 missense mutations that this patient presented. Then they are run through the prediction pipeline, and then you can see that about 25% of those missense mutations can be presented, in this case, either by A2 or B7. And then we come in with the transcriptome data, and we ask how many of those proteins that encode these missense mutations are expressed, and then we get a drop of about 50%. And at the end, for this patient in particular, we had 376 mutations that could potentially encode for candidate neoantigens. So using a curated approach as to the variant frequency of these mutations, the expression of these mutations, the affinity of those peptides, we selected 12 peptides that then we incorporated into a vaccine. And what I'm showing you here is actually, in this case, there were, this patient got 9 peptides. Each one of these is one different somatic mutation that is present, and that 9 mers were made, and they were incorporated in the vaccine. At the top, I'm showing you blood before the vaccine. At the bottom, I'm showing you after the vaccine. And what we see is that out of these 9, there were 2 neoepitopes that were pre-existing, and when we give the vaccine, we reveal 5 more. So the vaccine have allowed you to see the immunogenicity of 5 out of the 9 peptides, plus the 2 pre-existing. And this is an in vitro culture, okay? The cells are cultured for 10 days before we do the multimer assay. And then, of course, it's important to see that the cells bind multimer and that they can be expanded, but how about their functionality? So what I'm showing you at the bottom is chromium release assays, where we have either used an artificial antigen-presenting cell, and we have pulsed either with a mutated peptide or the wild-type peptide. And what we see is that only the wild-type peptide in all the situations is presented. So these T cells have high exquisite specificity for that one residue that has been changed. When we look then as to whether those epitopes are processed and presented, because this is critical, the cells need to process and present the epitope for effective lysis of tumor cells, we can see again that out of the 6 that I'm showing you here, 5 of them can really present the can really recognize the mutated peptide, but not the wild-type peptide. So in these patients, then there were 7 out of 9 responses that were detected, and all of those out of the 7, 6 could recognize processed and presented antigen, and none of them cross-react with the wild type. So can we see these responses directly from the blood? Because what we are using is a culture system, because we know from our experience from previous vaccines that that's the most sensitive method. But we're always asked, oh, can you see them in the blood? And that's what I'm showing you here. So this is actually before the vaccine. This is after the vaccine. And in this case, this was a patient that only received 3 neoantigens. And this patient was A2, so also received 2 share antigens, in this case, GP100 derived peptides. And what we see is that before the vaccine, we don't see any response to the neoantigens or to the share antigens. However, after the vaccine, we can directly detect on the blood up to about almost 1% those neoantigen T cells and some of the share antigens. And then I'm just showing you here flu M1 as a control that has not changed pre or post-vaccine. The other question that we're usually asked is, well, can these responses last? And what I'm showing you here is, again, another patient. This one got had a response to 3 neoantigens. This is right after the last vaccine dose. And then we look 4.5 months later and 8 months later, and the response is there. So we think that by giving these IL-12 producing mature dendritic cells pulsed with the peptides, we are generating a long-lasting memory response. And actually, I don't have it here, but we have some of these patients now have been 4 years after the vaccine, and you can still see these responses if you culture in vitro. And then last, this was one of the patients that received the vaccine. So it was a 54-year-old male who had melanoma, had surgically resected his lymph node and some of his meds. We used these to design his vaccine. He went into EP, and then he had a stable disease. Then we treated with the vaccine. And then some months later, after the vaccine, he progressed, so he went into PD-1. And then he was treated with anti-PD-1. And I just want to show you here that before the vaccine, he basically had no neoantigen response. We vaccinated, and he had a significant neoantigen response. And after the vaccine plus PD-1, that response was enhanced. But what is important here is that those lesions that the patient had after the vaccine with PD-1, they went away. And this patient now has been disease-free for 4 years. So this actually made us change our vaccine trial. And the way we're doing the vaccine trial right now is patients get vaccinated, and then 6 weeks after the last vaccine, they go on and get a cycle of PD-1. And we're monitoring those patients currently. So what lessons have we learned then from these 7 patients that we have done? We have actually used, to date, 60 short peptides that are optimal for the allele of interest. Out of those 60 short peptides, pre-existing immunity was only present to 8 of them. However, the vaccine elicited responses to 19 more, so almost half, and 33 of them are non-immunogenic, and this is the big question, why these are non-immunogenic. The only thing that we see that the immunogenic peptides have in common is that most of them tend to be very high affinity antigens. They tend to have predicted affinities of less than 50 nanomolar. and that has been confirmed experimentally. But we don't see that there is any positional bias. The mutation could be in the anchor residues that allow the peptide to bind to the MHC. The mutations could be in the residues that are actually pointing towards the TCR. So we still don't have a complete picture of what makes a peptide immunogenic and what makes a peptide non-immunogenic. What we do know is that our vaccine increases the antigenic breadth of the anti-tumor response, because you can see de novo responses that were not there. This is something that we have seen, that Catherine Wood has seen, that Ugur Sahin has seen. So the groups that are working in neoantigens have seen this increase of antigenic breadth that the vaccine promotes, whether it's a DC vaccine, a long peptide vaccine, an RNA vaccine. So we all agree on that. So there, we think this is important, again, because of tumor heterogeneity. The more antigens you target, the better it is. And in the case of our vaccinated patients, 6 out of the 7 are still alive and have no evidence of disease. So I'm going to change now a little bit from the vaccine to looking really more at the neoantigens that the vaccine has elicited. And I'm going to go back and talk a little bit about cold and hot tumors. So the initial observation from Tony's lab was that you could see a dichotomy between progressors and responders because of the infiltrates that you could see in the tumor. I think now we are starting to understand that it's not only the quantity, but it's also the quality of the T cells that are present in the tumor. And this is actually from a recent paper by Tom Schumacher's group, where he looked at 2 ovarian patients and 1 colorectal patient, and and 1 melanoma patient. And based on that, he proposed that it's not only the quantity, how many T cells you find in the tumor, but it's also the quality. It's how many of the T cells that are present in the tumor are actually tumor reactive, and how many of them are actually bystander. And there is actually a study from Newell that have actually looked at the composition of T cells in the tumor, and he reports that many of these bystander T cells are viral. So we might be misled as to how many T cells are in the tumor and how many of them are actually targeting the antigens for clinical efficacy. So we wanted then to look at T cell quality. We wanted to look at the antigen specificity of the T cells that were neoantigen in the tumor. We wanted also to look at the clonotype diversity. How many TCR clonotypes do we find that are against a specific antigen? And we wanted to look at the functionality. So it's not only to know that it's inflamed, but to know that it's inflamed, and what is inflamed here is actually having some efficacy at destroying the tumor cells. So before I talk to that, I'm going to just very quickly talk about tumor genetic heterogeneity and the neoantigen landscape. And as I already alluded, when we look at the range of a spectrum of mutations, mutational burdens between tumors, there are those that are very low, there are those that are very high, and melanoma, it's at the high end, so it has the highest mutational burden. However, when we look how those mutations are distributed within the tumor, we see that there are basically 2 kinds of, well, there are many, but to make it simple, let's say there are 2. There is mutations that are truncal or clonal, that they arise very early during the tumorigenesis process, and they're present in all the cells, all the tumor cells. And then there are those mutations that arise late during tumorigenesis, and those are considered branch or subclonal. So not all the mutations play the same role, and not all the mutations are the same as target antigens, because as you can imagine, it would be better to target these clonal mutations than these subclonal, because these will target all the tumor cells. However, when we look at melanoma and we see how heterogeneous melanoma is actually the less heterogeneous of tumor antigens. So if you think of heterogeneity, tumor heterogeneity as a tree, melanoma is a palm tree. It has a long trunk with a few branches at the top. So we wanted then, knowing the neoantigens that we had identified in our patients, could we go back and look at mutational burden, heterogeneity, and T cells, and try to understand how all these things fit together. So we had basically 3 questions. What is the melanoma neoantigen landscape in patients for which we have multiple tumors? The second question was, what is the intratumoral distribution of the neoantigen-specific TILs? And then what is the correlate between the neoantigen landscape and the neoantigen-specific TILs? So again, very quickly, we use this pipeline to identify neoantigens. And then I'm just going to use the same patient that I shown you before, where we had 375 potential neoantigens. So we were interested first in defining the landscape of these neoantigens. And in this case, we thought this patient was worth studying because we had 6 different tumors from these antigens. So we could ask, what is the distribution of those 375 neoantigens in these 6 tumors that were taken at different times during his treatment? And some of them were taken before EP, before RAF inhibitor, before RAF-BEC. So, and then all of them were before the vaccine. So we took these 375 mutations, and we did targeted sequence, very deep, so that we could get over 1,000 reads for each one of those mutations. And then in collaboration with Malachi and Novi Griffith at Washington University, we use a program called Cyclone that allows you to organize those mutations according to the variant frequency in clusters and ask the question, are they clonal, are they subclonal? And what we see is that when we use these 2 tumors that were the ones that were used for the vaccine, we have basically 7 clusters. Out of those 7 clusters, there are basically 3 clusters that are shared by these 2 tumors, while there are 4 clusters that are unique. And that out of those clusters that are shared, we have some that are clonal and some that are subclonal. When we then extend this analysis and we look then at the other 4 tumors, and this is a pairwise comparison of all the 6 tumors that we have, what we see is that, just as it was reported before for melanoma, there is little heterogeneity between melanoma tumors, because most of them are shared by all the 6 tumors. And more important than that, when you look at the neoantigens that were targeted by the vaccine, they're all shared and they're all clonal, and one is overexpressed. So there is little heterogeneity. Although these tumors went through different therapies, there is little change in the clonal composition. So we thought, okay, so now we have what is the neoantigen landscape in these tumors. Can we now take our neoantigens and go back and ask, do we find them in all these tumors? So we used the ProImmune Reveal and ProVench to actually make 60 pentamers that represented the best and highest affinity of the neoantigens that we had identified in these patients. And as you can see here, these were all really high quality, high affinity neoantigens, and went back and looked in the TIL population and asked, how many of them can we find out of those 60, which represents about 20% of the 375 neoantigens that we had said were candidates. And I'm just showing you here a few of them, and I can tell you in all manipulated T cells, out of the 60 neoantigens, there was only one that we could see. And that's this one right here. And we have 2 different tumors. When we then take these TILs and we grow them in culture for 10 days with all the 60 peptides, and we add how many of them can we see, then I'm showing you down here, just forget, this is the blood. You couldn't see in the blood except 2. But in the TILs, you could see this one here, and you could see 6 more. So again, out of 60, you can only see 7. So this goes in agreement with everybody that has been working on neoantigens and find that the neoantigens are very rare. But what this allowed us to do was to have, we had 7 that came from the vaccine plus 5 that came from the TILs. So we could ask for 7 and 5 is 12. We could ask for 12, we had 12 bona fide neoantigens that we could go back to the tumors and ask how many of them can we find in the tumors using a method more sensitive than cell culture. And to do this, then we prepare what we call neoantigen-specific TCR libraries. So we grow the CD8s in the presence of the mutated peptide. We make cell lines. We sort these lines using multimers to 90%, 99% purity. Then we subject them to TCR sequencing. Then we pick any clonotype that is more than 0.1%. And then these libraries are then used to go back in the unmanipulated either CD8s or tumors and ask how many of these clonotypes can we find. And to validate this approach, we have now gone on and cloned TCRs from each one of the antigens that we're studying, and I'm just showing you 2 of them here. This is just this neoantigen where we isolated an alpha and beta TCR. We put it in jerkats, and we went back and asked, can they recognize the mutated peptide or the wild type? And you can see that there is only recognition of the mutated peptide, and so is the case down here. So these neoantigens libraries allow you to, on a molecular method, go back to the tumors or to the blood and ask how many of these neoantigens we can find and how many of the clonotypes we can find. So we go back to this same patient, 66. I just show you this, showing you that the neoantigens are present in all the 6 tumors that we study. However, when you go back and ask how many of the TCR clonotypes can we actually find, what we see is 2 things. Let me just walk you here. Every one of these dots represents a T-cell receptor. So these tumors are all highly infiltrated with T cells. So there are lots of T cells in each one of these tumors. However, when you look at the clonotypes to the different neoantigens, and here when it says pre and post, this is the vaccine. Pre-vaccine and post-vaccine. So one thing the vaccine does is for each neoantigen, it increases the TCR clonotype diversity. So you go from having maybe 2 to having 5. And here, I can't remember, I think we went from having 9 to having something like 17. So the vaccine not only increases the antigen diversity, but also the clonotype diversity for each one of the antigens. And then you take these clonotypes and you go back into the tumors and you ask how many of them can I find. And each color represents a different TCR sequence, and what you see is that in spite of being the tumors being highly infiltrated with T cells, there is only maybe 1 clonotype. So for each one of the neoantigens, not only you can find few neoantigens, but of those many clonotypes that are possible, you can only find 1, at most 2, and in some cases you can find none. So the antigens are there. There can be a high diversity to it, but prior to vaccination, you see none. And that this is not unique to this patient. I'm going to show you a second patient. In this case, we had 3 tumors from this patient. Again, the vaccine induces an increase in the TCR clonotypes, but in the tumors themselves, you can find one or two, and in some cases, none. So these neoantigens are there, but unless you prime them by vaccination, there are no immune responses present. And this is not unique to the neoantigens, because when you look at shared antigens, in this case GP100, and these patients are A2, again, the vaccine increases the diversity, but if you go back to the tumors, and these tumors were all GP100 positive, nothing. So again, quantity does not mean quality. So for these, we have had now 27 neoantigens that we have done this type of analysis, anywhere from 2 to 6 tumors per patient. And in the majority of the situations, what you get is something like this. Either the tumors are bare of T cells that are specific for the neoantigens, or you find maybe one. So in summary, despite sharing of clonal neoantigens, because all these neoantigens were clonal, like I show you, the CD8 repertoire tends to be small and tends to be monoclonal, or tends to be absent. So this really extends and redefines the concept of cold tumors. So when we talk about cold tumors, we not only should think about cold tumors as those that have no infiltrate, but also those that have infiltrates where no T cells specific for tumor antigens are present. So we now are looking at viral responses too, to see how many of those T cells that are there are viral, and what is the percentage between tumor-specific and viral antigens. We think that, at least in melanoma, part of the reason there is an ineffective T cell immunity is because there is immunological ignorance. This is a term that was quoted by Sincronago many, many years ago, and it means that the antigen is present, but for reasons that we don't completely understand, is not present, is not priming an immune response. And we think this immunological ignorance is happening at two levels. At the antigen level, because we can see new antigens that are revealed by the vaccine, and at the TCR clonotype level, because we have a very few clonotypes, but we vaccinate, and then we see, 6, 7 more, or even more. So we think that when we think about checkpoint inhibitor treatment, vaccination is a good therapeutic avenue that would allow us to increase both the breadth and the diversity of the neoantigen response, because the T cells are there, but are at such low frequency that unless you prime them, they will not have clinical benefit. And then I just want to finish by acknowledging the people that have contributed to this work. Gerald Lynette, who is my lab partner and is the oncologist in our lab. Then our lab, primarily Lorea and John Chu, who did most of the characterization of the T cell responses. Are members of the Center for Cellular Immunotherapy that do the regulatory aspects of our trials, that do the manufacturing of our vaccines. All the TCR work was done in collaboration with the Human Immunology Core at Penn. Elaine Martis and Vince Magrini, with whom we started these studies to do all the genomic aspect of our project, and now a group at Washington University, the bioinformaticians that allow us to predict neoantigens and are so funding. I'll be happy to take any questions. Thank you. Sure.
Do you have any sense of why some of these are more immunogenic than others?
I wish I could tell you. No. No. We have,
It's a challenge across the field, so.
yeah. The N is still too small. It's not, we can't find anything that correlates in terms of, like I said, expression, affinity, whether they're clonal or they're subclonal. We don't understand enough about immunogenicity yet, I guess.
So great talk. Thank you for sharing all of this. I did have a question about, you have looked at just the CD8s and the class 1. Would there be value in looking at the class 2
So,
and
yeah.
CD4? Could you improve that?
Yeah, so
diversity?
when we modify our trial to include PD-1 after the vaccine, we also modify it to include 2 CD4 epitopes. And we have 3 patients that we have done that. We have gotten some CD4 responses. Out of 6 CD4 peptides that we have included, we have gotten 1 CD4 response. And again,
All right. Yeah, maybe we can discuss offline because we took Catherine Wu's paper and what she recognized and put it in the looking for CD4s, and we did find we could improve the diversity or coverage just by including. So something we can discuss later.
yeah. So I should say that in our vaccine, because we always are always like, oh, you don't have CD4 help. When we make our vaccine, we actually, the first dose of the vaccine, the diseases are pulsed with the flu vaccine. So the flu vaccine provides help for that CD8 generation.
Right.
But we're now including CD4 epitopes because we think we would learn something from that. But that's all I can tell you right now.
And can I ask one more question? Going back to the first question about why some of them are antigenic or why, so is there a role of what we have as a T cell repertoire and
Sure.
tolerance?
Absolutely, yeah. So we're actually now taking these 60 peptides. It's a lot of work, but we're taking these 60 peptides, and there is something that people call outsourcing, which is basically you go to healthy donors and you use these neoantigens. Schumacher has done it. You go and you see how many of them you can fish T cells out of normal donors. And we think maybe by doing that we'll learn something about what is immunogenic and what is not. But it's a lot of work because you need to do a lot of donors to see. So, yeah.
Thanks.
Okay.
Question is, you have shown some of these T cells have higher clone numbers following the vaccination. Have you characterized what type of CD8 cells they are, what markers their activation because that is what it boils down. You can have a clones, and if they are not responsive, if they are not signaling properly through 4-1BB or other things, or they express TJ or PD1 or all sorts of things and communicating with regulatory cells, then it doesn't matter. You could have a clones and you could see them because of the tolerance and autoantigens, all these things. So you need to have at least any idea what type of CD8?
So right now, because we know what TCRs they have, and we know what their antigen specificity is, we're doing some single cell analysis, doing both TCR and transcriptome at the same time, so we could see what their transcriptome, their gene expression pattern is, but I don't have anything to
I guess related to the first 2 questions, and I have a lot of ignorance of the field, but is there any relation between the types of amino acid mutations and their ability to drive immunogenicity? So by that I mean, a glycine to an alanine would be a minor switch, whereas going to a lysine would, whether charge or hydrophobicity, if that has any role in driving these responses and whether or not something's immunogenic.
It has been suggested that hydrophobicity is good for antigenicity. I don't know we can say. What I can tell you is that every allele, every HLA allele, it's a case in itself. So the rules that you might learn for A2 are going to be very different from the rules that you might learn for B8. And then there are certain alleles like A3 that I particularly do not like. Because I don't know what's the deal with that allele, but it is so hard to get responses to. And good affinity, good binding, good stability, and
Yeah,
so
but I guess beyond the affinity to the actual HLA itself, because that's part of your pipeline. I just
the
mean
peptide itself.
the peptide itself, in terms of its affinity for the HLA, that's a separate issue. But I
Yeah.
wonder if the magnitude of the change in what is exposed to the T cell after you've already predicted whether or not something binds,
Yeah, I mean, that's a good
which
point.
might hold true across HLA.
Yeah,
subtypes.
that's a good point, one that we have not looked at.
Generally, it was thought where the anchoring the peptide chain is there, it will make it more.
Yeah, I mean, the other,
The evidence.
yeah, I mean, the other thing is, you know, it has been proposed that the more different the mutated peptide is from the wild type, maybe the more antigenic it is. I have to tell you that I don't think that is true, because when we look at our peptides again, and then it's small, but you can have the wild type and the mutated have about the same affinity, and it turns out to be the one that gives you, 60 different clonotypes when you use it.
So Beatriz, thank you very much. That's really interesting. You mentioned just then, going back to your A3 question or issue, do you see, because you obviously can't generate those responses to A3, is that also similar for A11 positive patients that have quite a similar?
No, A11 is a good one.
It's a good one.
Mhm.
Okay, interesting.
Yeah, that's it. And that puzzles us too, because they're so related.
Yeah, yeah.
They even share peptides.
Yeah, absolutely.
And it's like A11 is good, A3
Okay,
is
well
no good.
that thrashes that hypothesis. But did you also see then, from obviously because that's in vitro trying to generate those responses, is there any correlation between A3 positive donors and response rate overall in regards to tumor outcomes?
We have never looked at that.
No.
Yeah.
Okay. And the other question I had was just in relation to, obviously, interesting picture about the trunk length and the kind of clonal mutations versus subclonal. So obviously, you kind of hit it lucky with regards to the melanoma. Obviously, you've got large diversity and you've got that a lot of early mutations. Does that also mean that what are the knock-on effects for applying your strategy in other cancers where it's much more subclonal mutations?
Right. So we are initiating we have initiated a trial in colorectal where there is a lot of heterogeneity. So there, when you do the sequencing, what you find is that there are very few truncal and a lot of, so there what the approach we're taking is with it just targeting a lot of subclonal, and we'll see what happens. Yeah.
And what is it about melanoma that makes the mutations I mean, I just don't understand, but that makes the mutations happen earlier on in the process?
That's a question for Jerry more than for me, but I think what happens is BRAF is a good driver, right? But because of its zone exposed area, you have many years before that transformation happens, that you have all these passenger mutations that have been accumulating because you're getting zone exposed. So a lot of those C to T transitions are happening, but I guess it's by the time BRAF mutates, then you're carrying those passenger mutations along with the driver, and it looks like it's very little. I think colorectal is probably different, but that's me thinking.
Yeah, so I actually had a question that was similar to Ryan's, so I'll make it like a follow-up comment very briefly. With the influenza peptide from the Magix, the 5866, which is immunodominant for the A2 allele. what we did was to actually take the valine, which is in the middle of the peptide, it's supposed to be one of these TCR contacts, and then we started looking at how the TCR repertoire changes as a function of changing the, that valine, right? And we started from more conservative mutation, and then work our way out of the, the physical chemical, range of that valine, and then make it a charged or bulkier, or somehow make it different and see how that would affect the repertoire. And so you could actually build the network of TCR, of T-cell clones based on their repertoire, right, based on their sequences, and see that the one that was responding to the valine, for instance, would also respond to the leucine, and would also respond to an isoleucine, and then maybe would not respond to the lysine, right? But the one that responds to the lysine would respond to maybe the isoleucine for another reason, but not the valine anymore. So you could actually construct that network and sort of generate, the overall scenario of how the different T cells are elicited or not, based on the nature of the of that individual residue they are mutating. And so possibly one of the reasons why you are seeing more or less immunogenicity is potentially, not only how well they bind to the MHC, but also what's actually pointing upward. And so now you're eliciting some of these clones, or maybe not, depending on how the repertoire is actually constructed in these patients.
Yeah. I'm just hoping that as we all vaccinate different ways, but also we get enough database that we can then start looking for patterns. I think it's too early now. Yeah. Thank you.